Hello and good evening, everyone. How are you all doing? Am I audible? Is my voice loud and clear? All right. So, are we all ready to do another very interesting topic? A topic from your plant science? So, today is day 18, and we will be talking about photoperiodism and vernalization. A very, very interesting topic. You see, we always, whenever we think of light, sunlight, and plant, we always tag that with photosynthesis. But that is not the only place where sunlight is required. There are many other things, many other physiological functions which are carried out because of the sunlight and also because of the darkness that follows after the day. Have you ever noticed uh, something at the back of your yard, in your garden? If you have placed a pot or say any kind of a thing, okay, a board, a pistboard or something on the grass for several days, when you remove that flower pot or when you remove that tub or that cardboard, you will find all the grasses, they have paled and thinned and they are just not looking like that of the other grasses. Have you ever noticed? How many of you have noticed this? Those who have a garden will definitely come up with this kind of an experience. Is there anyone who has noticed such kind of a situation? Yes, Jeffrey has. So what happened over there? The grass they are surviving, right? But they are not growing. They are not healthy. So all of this for their healthy growth, for their for their survivability, for all these we need sunlight. Today we will be mainly focusing on these two topics and we will start with the definition. So first and foremost, photoperiodism. This is defined as a response to changes in day length that enables the plant or any other living organism to adapt to seasonal changes in their environment. So this is a word which we can apply to other organisms also, but since we are talking about the plants, plant sciences, so we are going to focus our entire discussion on plant science. Okay, so which enables the plant. Now this uh, photoperiodism is best studied in the flowering of the plants. But not only flowering, it is also involved in the dormancy, in bud dormancy. It is also seen when you have the bulb or the tuber initiation. So these are the other physiological processes which are controlled by this photoperiodism, which is nothing but the response to changes in daylight. Then what is vernalization? Vernalization is promotion of flowering at subsequent higher temperatures brought about by exposure to cold. This is the key word. Brought about by exposure to cold. So when we talk about vernalization, we will see how cold temperatures can induce flowering. Now, apart from the temperature, other signals such as total light radiation, water availability, these are also important cues for vernalization. Okay? Now, let us first go through some basic things, basic informations regarding photoperiodism. First and foremost, during the spring and autumn periods, day length alone has an ambiguous indicator of time of the year. Understand this. We have the calendars, right, to understand what is the time of the year. But these plants, they obviously don't have a calendar, right? But they have to produce flowers at a particular time of the year. So there has to be some external signals which is working, which is being perceived by the plants to understand what is the time, what is that day of the year. Okay. Now, not only the day of the year, but also the day length it has to measure. So, the progressive changes in day length provide a certain environmental signal for the passage of the seasons. Why? Because we all know that summers, the day length is different, for winters, the day length is different. So, if you can measure, if you can sense this day length, you will definitely understand what is the time of the, what is the season, what is that month of the year. So in order to locate the time of year accurately, a timekeeping mechanism in photoperiodism operates, and this mechanism has to operate with precision. It is a must. Okay, so it is, it is a must to be insensitive to unpredictable variations in the environment. And one of the most unpredictable variation is your temperature. So this timekeeping mechanism should not be affected by any kind of these variations of the environment. Otherwise, we will be getting wrong information. We will be sensing wrong information. So this is very, very crucial, accurate. The rate of change of day length varies during the year, being fastest in the spring and autumn. Next, so if it is fastest during spring and autumn, then you can definitely understand the seasonal range also. So the seasonal range and rate of change of day length is lower in the tropics than at higher altitudes. Sorry, higher latitudes. So, keeping these things in mind, this mechanism has to work. Okay. So, as we see here, this is the day length and the night length that is showing here, and this is showing you the percent of flowering. So, this is a photoperiodic response in long and short day plants. The critical duration varies between the species. So for long day plants, you need longer days. From the word you can understand, I will go into the details in the next few slides. And for the short day plants means you need shorter day length but longer nights. All right. So you reach a peak where, which is your critical period. If you can't reach that peak, whichever plant you are, whether it be a long day or a short day, there is a critical peak. Then flowering will not happen. So let us understand what are these short day and long day plants. It was first the American physiologist Gardner and Allern who were the first who saw clearly the flowering and this many other responses in the plant which could be accelerated either by these long days or by the short days, and that completely dependent on the plant. So they were the one to introduce the word photoperiod. Photo, okay? Photoperiod. They were the one to introduce. And also the word which follows photoperiod is photoperiodism. So according to them, there are four different types of plants. One is a short day plant, which are those that flower or in which flowering is accelerated by days which are shorter than a critical day length. So I told you, no, in the previous graph I showed you there is a critical period. Okay, so here the flowering is accelerated by days which are shorter than the critical day. Examples can be rice and soy. Whereas in contrast, the long day plants, they are the plants that flower or in which the flowering is accelerated when the daily light period exceeds the critical day. And good examples can be the wheat, the barley. So that's why they are known as long day plants. Now, apart from these two types, you also have the short long day plants. So these are the plants that flower only after a sequence of short days followed by long days. So it is not like you have, if you give a prolonged long day exposure, then they will flower. No, there has to be a balance between the short days and the long days. Okay, so you can say that these are the plants which are mostly flowering during the spring or the autumn where a transition is taking place. Okay, and example can be the white clover, Canterbury bells. So this is the example. This is your white clover. Next, you have the day neutral plants, which are the plants that flower at the same time, irrespective of the photoperiodic condition. So, no matter whether it is a short day or a long day, it will flower. So, these kinds of plants they are mostly present in the tropics. Okay, where the day lengths they don't vary much. There is very less variation in the day lengths. So examples can be the kidney beans, which has evolved near the equator where the day lengths are constant. And also you can have the desert sand verbena flower. Whenever there is water, it will flower. Okay, it's not depending upon the day, or it's not dependent upon the day length. So these are the four different types you have. So accordingly, if you see, this is a set of your short day plants. If this is your critical, if as you can see this dotted line, if this is your critical duration, then look at this very carefully. You know, these are the graphs which you need to study very thoroughly. Here what you are giving is a longer day exposure, longer than the critical day. Okay, and this is the night. So you don't find any flowering here. In this case, you have a shorter tail end, shorter than the critical period. Okay, so that's why you find this plant flowering. Here, what you're doing, you are giving a shorter day, shorter than the critical period, but you are interrupting the dark phase with a flash of light. In that case, again, there will be no flower. Alright? So that means the dark phase is very, very important in case of short day plants. You cannot tamper with the dark phase. Okay, it has to be of a proper duration. Next, you have the long days. So, when you expose it to longer days, day lengths greater than the critical day length, you will see flowering. If you have shorter day lengths, shorter than the critical, there will be no flowering. Now, see, these, so from here it is very clearly understood that these plants they need longer exposure to light, right? So, in that case, if you give a shorter day exposure, but you interrupt the dark phase with some light, then also these plants are seen to flower. Okay, because they are getting exposure to light, and that is what is triggering their flowering. Now, not only you can divide these plants under these four heads, but you can also divide it or subdivide it under obligate and facultative. Obligate or qualitative types, where the particular day length is essential for flowering, or it can be a facultative or quantitative type, where the particular day length accelerates but not essential for flowering. Okay, so again, that depends from plants to plants. Next, so now the question comes is, how does the plants monitor the day length? Is it that they are monitoring the day length, or is it they are monitoring the night? What is it? So it has been seen that in the case of your short day plants, the flowering is determined by the duration of the darkness. What we have seen in the graph also, if you interrupt the dark phase with light, it will not flower. So it's very clear that the dark phase is very, very important in case of short day plants. So they can flower under longer than critical day length provided it is followed by a sufficiently longer nights. So you have to maintain the ratio. Now in case of your long day plants, since they require a lot of light, even if you provide short days, if you follow it with shorter nights, that means you interrupt the night with some light, as you have seen in that graph, then they will flower. Okay, so the dark period is essential for flowering. So again, there is a no representation of the short day and the long day. There are different conditions. As you can see, for in this case, shorter day, shorter light, longer darkness means for SDP it is flowering. For a long day plants, it will only be a vegetative growth. On the other hand, if you have a longer day, shorter nights for the short day plants, it will be vegetative. But in the case of your long day plants, it will be flowering. Now, in case of your this short day, but you're interrupting the dark phase with some light. This is beneficial for whom? For the long day plants, not the short days. Now you are increasing this exposure to light for a longer duration. Okay, you're interrupting the dark phase with light for a longer duration. This again will be beneficial for long day plants. You're giving almost equal amount of light and dark. Then also you will see that some of the long day plants are flowering. But these have no impacts on the short day plants. It will only be a vegetative growth. Again, short day will be followed. Under short day plants, you will get flowering when you have longer light, but also followed by sufficiently longer dark phase. Okay, so one thing is very clear, for short day plants, dark phase has to be longer. Okay, so night breaks can cancel the effect of dark period. So night break treatments of only a few minutes are effective in preventing flowering in many short day plants, including xanthium, Forbitis, but much longer exposures are required to promote flowering in long day plants. So this is the graph. Okay, so this is the time of night break from beginning of the dark period. What you find? This is for the short day plant. If you give the light period, if you expose it to the light period, then this flowering is reduced. But in case of your long day, if you expose it to the light period within the dark phase, then the flowering is good. Okay, so there are various representation of the concept, and you should be very thorough with these representations. Now, in the first slide itself, we have mentioned that you not only have to measure the seasonal change, okay, but you also have to measure the change during a day, right? Because until and unless you can measure the day length, you will not be able to measure the seasons. So therefore, the circadian clock, this is involved in the photoperiodic timekeeping as well. What is circadian clock? Can someone tell me what is circadian clock? Anyone who has the idea of circadian clock? Come on, let's see. Circadian clock. This is a word which can be applied to your animals also, animal behavior also. Circadian clock, the sleep and the wake movements. No one? Alright. Circadian clock is an internal time clock that is there, which is measuring your day length. Okay, and this internal time clock, this is controlled by the light. Light has got an influence on this uh, time clock, and accordingly, the plant or the animal modifies their behavior or shows that particular behavior at that particular time of the day. Okay, like where how does the sunflower knows when the sun is up? All right, or you must have seen the plants, especially the flowers. They will bloom at a particular time of the day, and then again they will, shrink when it is evening. The leaves, they will stretch when it is day, but they will again start shrinking at during the evening. From evening onwards, they will start shrinking. Have you ever noticed that? If you have a flower pot at home, you will see. If you haven't noticed, from today onwards, like from tomorrow onwards, notice that. These are all the circadian clock which is controlling. So, according to the clock hypothesis, photoperiodic timekeeping depends on an endogenous circadian oscillator of the type involved in the daily rhythms described in relation to phytochrome. So, why am I saying oscillator? Oscillator means it has to undergo some oscillation, right? Because the rhythms, okay, or this particular behaviors, it follows a rhythmic pattern. Okay, it sometimes peaks at one point of the day, again it goes down, again it peaks at a point of the time of the day, again it goes down. So that's why we are saying it is an oscillator which is synchronized with the light. So what happens here is, if for this flowering, okay, if you can expose the light, again the long day plants, to more of light, you will find flowering. All right, because you're increasing the light sensitive phase. Got it? So next, how does the plant? All right. Uh, Before I go to this, one more thing I need to mention here. One example of this circadian clock can be your soybean. Okay, a good example is your soybean clock, and it has been seen that when they are transferred from an 8 hours light period to an extended 64 hours of dark period, the flowering response to night break shows a circadian rhythm. So flowering in the short day plants, they have shown to respond both a dark period of sufficient duration and a dawn signal at the appropriate time. If you don't expose the plant to the dawn signal at the correct time, then flowering will not be happening. There will be no flower. All right. So that is very, very important. You might experiment with the time duration. Doesn't matter. Two things are very important. One is the duration of the dark phase and the dawn signal. Because this acts as a trigger. So, what is that part of the plant that receives this this stimuli, this photoperiodic stimuli? From several experiments, it has been shown that leaf is the one. Leaf is the site of perception of the photoperiodic stimulus. So, in response to photoperiod, the leaf transmits the signal that regulates the transition to flowering at the shoot apex. Okay. Now, the photoperiod regulated process that occurs in the leaves, resulting in the transmission of the floral stimulus to the shoot apex, it is referred to as photoperiodic induction. And all of these things has been shown, or it has been discovered, or you can say it has been proved from grafting experiments. Graft, do you know what is a graft? Who can tell me what is a graft? Have you heard about grafting? So we take, if this is one plant here, we will take, say, the shoot part of this plant and graft it on another plant which has got only the root part. This is what is called as grafting. Okay. So from several of these grafting experiments, these theories have been proved. And it has been shown, very interesting, that photoperiodic induction can take place in a leaf that has been separated from plant. So it's not mandatory that you have to keep the leaf attached to the plant. So from this observation, we can definitely conclude that it is a leaf which is perceiving the stimuli. Okay, like for example, you have this short day plant Perilla crispa, where if you take the excise leaf and expose it to the short days, then it can cause flowering when subsequently grafted to a non-induced plant maintained in longer days. Okay, so what you do is, say this is your plant which has not been induced. Okay, this is being exposed to long days. You take a leaf of this plant, just expose it to the short day lengths. All right, expose it to the short day lengths. And then you graft this leaf to this plant. You graft this leaf to this plant, and you will see that this plant has started to flower, produce the plants. So this result, what it can indicate, that photoperiodic induction depends on events that take place exclusively in the leaf. Now the next question was, if it is produced in the leaves, okay, and when you're grafting it on another plant, your flower buds are, say, somewhere here, then from the leaf, how the signal is coming to the flower bud? There has to be some pathway. So it was first hypothesized that maybe it is using the phloem. Later on again, With several experiments, it was shown that yes, definitely, it is a phloem which is transporting the signal. Okay, so once produced, the flowering stimulus appears to be transported to the meristem via the phloem. It appears to be chemical rather than physical. So this flowering stimulus, no, what I'm referring to here, this is a chemical entity. It is a chemical thing. Okay, not a physical thing. And this has been actually shown that like it is following it is taking the help of the phloem. This has been proved by the experiments which involves girdling or localizing heat killing, okay, or the preventing the movement of the floral signals, all other methods. So via those methods, it has actually been seen that if you say, suppose here put a, you girdle this section, then the signal will not be able to transmit to this end. Okay. So the rate of transport of the flowering signal is comparable to or somewhat slower than the rate of translocation of sugar in the phloem. This is a takeaway point you can keep in mind. So here also the signal follows a source and a sink concept. So for translocation of your sugars, we have seen there is a source and sink concept. For your transferring or transportation of this flowering stimulus, there also a source and a sink. So this will be your source, this will be your sink. Okay. Now, because the floral stimulus is translocated along with the sugars in the phloem, it is subjected to sourcing relations. Non-induced leaves position between the induced leaf and the apical bud will tend to inhibit flowering by serving as preferred source leaves for the bud. That means, suppose this is your plant, here you have a bud. Okay, these are the branches. Here you have a bud. Now, you have placed a non-induced leaf here. Okay, and you are grafting a induced leaf somewhere here. So this is your induced leaf, this is your non-induced. From the induced leaf, what will happen? The floral stimulus will try to go to the bud, right? But this bud, since this leaf is nearest, so what you will get is a negative signal. Hence, even though if you place the induced leaf, the bud will not flower. The positioning of this induced leaf in that graph is important if you want to stimulate the floral induction. Clear? Now coming to the phytochrome, which is the primary photoreceptor and photoperiodism. Phytochrome, we all know, they are the pigments which is receiving the light. Yes. So in short day plants, a night break becomes effective when the phytochrome PR converts to PFR. PR means it is a phytochrome that absorbs red light, and the PFR means it is a phytochrome that absorbs far red light. So if there is a night break, it becomes effective only when the supplied dose of the light is sufficient to saturate the phytochrome conversion. Now, after this exposure, if you want to revert back, what will you do? You will expose it to a far red light. Okay. So, if you expose it to a far red light, what will happen? This PFR will get converted back to your PR. Okay. And if it gets back to your PR condition, that means what? It will will become the inactive. So in short, what I can write, PR exposed to red light gets converted to PFR, and this is your active form. This gives out the signals. But if you want to revert back, you have to expose it to the far red, which will be absorbed by the PFR and it will get converted to the PR, which is your inactive. Okay. Now that is the case of your short day plants. But in the case of your long day plants, a night break of red light, this promotes flowering. Okay. And later on, if you expose it to the far red light, then that will prevent this response. But in the case of short day plants, it will not show any response. So again, if you come to this diagram, this is showing you, this is for your short day plants. Okay. So uh, longer days, shorter nights, no induction. Longer nights, shorter days, induction. You are interfering the long nights with the exposure to red light. So that means there will be no conversion, no flowering. But however, if you follow two simultaneous exposure, one of the red light followed by the FR, far red, what happens? When you are exposing it to the red light, it is becoming active. Okay? When you are exposing to the far red light, then the phytochrome is becoming inactive. That means even though this is a light, it is inactivating the phytochrome. So therefore, this will be equivalent to darkness. Okay, this exposure will be equivalent to darkness. It will nullify each other. So therefore, there will be flower. Again, look at this. You are in, you know, you are exposing it to red. That means you are activating. You are inactivating. Again, you are activating. What will happen? No flowering, right? Because in that case, this will be equivalent to light, interruption of light, so no flowering. But again, if you can cancel the signal, okay, if you can turn it inactive and again make it look like it is the dark phase, then again it will show flower. Has everyone understood this? Raise your hand, tell me. Light bulb. Let's see if this light has made you understand this concept. Type LB, light bulb, quickly. Batti jali, have you understood, or you are still in the dark phase? So you can get questions like this. These experiments are important, otherwise the concept is very simple. You need to understand the experiments. You need to understand what is happening. So that goes with the short days. Look at the longer days. For longer days, long day, flowering. Short day length, no flowering. Long days exposure to red light, there will be flowering. Long days with exposure to red light and PR, no flowering. I hope you are following. Why there will be no flowering? Because this is equivalent to darkness, right? So it is not inducing it. And the day lengths are also below the critical level. It is not above the critical level. Therefore, it cannot induce any flower. So two things you have to see here. One is the critical level, whether it is crossing, it is beyond the critical, or is it less than the critical. And secondly, what kind of exposure you are giving here. All right. In this case, again, lower than the critical, but here you have given a red light. That means you have induced flowering for the long day plants. This is lower than the critical, but here you have inactivated the signal, so no flowering. The comparison is very, very important. Now you can do all of these modifications using the artificial light. So as we have already seen, far red light modifies flowering in some LDPs. So here what we have, the response is proportional to the irradiance and duration of the far red light, and is therefore a high irradiance response. Okay, so in this case, it is the phytochrome A which mediates a response to your far red light. Now, when the plant is exposed to far red light for 4 to 6 hours, flowering is promoted compared with plants maintained under continuous white or red light. This is very important, note it down. So, you can have a situation where you're giving longer days followed by some shorter nights. Okay, that will definitely promote flowering. But if you want to trigger this flowering process, the best way to do is give a moderate day length and then interfere the dark phase with the red light. Because red light is the triggering signal that will promote your flowering. So response mediated by phytochrome B, this does what? It reduces this exposure. Okay, so once you're exposing it to red, once you're exposing it to the uh, far red, okay, so this rhythm continues. Next, if you look into the blue light photoreceptor, this also regulates flowering, not only the red far red light, okay, but also the blue light. So in some cases, it has been seen that this long day plants like your Arabidopsis, this can be induced flowering with the exposure of blue light. So the role of blue light in flowering and its relationship to circadian rhythms have been investigated by using a luciferase reporter gene construct. I'm not going to the details of this. This is just for your information that these experiments are done with the help of the reporter gene. So in continuous white light, what you find is the cyclic illuminates has a period of 24.7 hours, but in constant darkness, the period lengthens to 30 to 36 hours. Now, at this time, if you give either a red or a blue light, then that will reduce this period to 25 hours. So, it is going to reduce this exposure. Okay. So what you are doing, what you are initiating with the help of the red light, you can do the same thing with the help of the blue light as well. So both phytochrome and blue light photoreceptor are involved in the period control. So next, moving on to your vernalization. So what is vernalization? That was about your photoperiods. I hope it is clear to everyone. Okay, so coming over to your vernalization. So photoperiodism was about perceiving the light, daylight, and inducing flowering. Vernalization is a process whereby flowering is promoted by a cold treatment given to a fully hydrated seed. So remember, in the first slide itself, we have seen that for vernalization, not only the cold temperature is important, but also some other factors like water, that is also important. So in this case, the seeds, only those seeds which are hydrated, can be initiated to flower if they are exposed to cold temperature, otherwise not. Okay, so dry seeds do not respond to cold temperature, and without the cold temperature, without this cold temperature, what will happen? Plants that require vernalization will show delayed flowering or remain vegetative. And in many cases, these plants grow as rosettes with no elongation of the stem. So if you look at this picture here, this is a winter annual Arabidopsis without vernalization, so it is forming a rosette kind of uh, morphology. Whereas if you induce flowering with vernalization, then this is the normal structure of Arabidopsis, what you get to see. Okay, so the form morphology will change. If it is not flowering, it will show vegetative growth. Now, before we go to some more concepts, let us have a quick concept check. The first question is on your screen. When the dark period of short day plant is interrupted by brief exposure of light, then the plant produces more flowers, will not bear any flower, turn into a long day plant, produce flowers immediately. What should be the correct statement? What should be your correct statement? Shivani, Bindu, Vidya, what happened? And others as well. So if you can answer this question, if you listen to this entire lecture completely, you know you will get the notes for your exam. So let's see how many of you have paid attention. Okay. The correct answer will be when the dark period of short day plant is interrupted by brief exposure to light, then the plant will not bear any flower. Alright, because for short day plants, you do need some, you need a longer day length. Next, Plant that require not less than 10 hours of light to flower is called what? The options are day neutral plant, short day plant, long day plant, and none of the others. So in this case, the correct answer will be long day plants. Okay. So moving on with your vernalization. So vernalization results in competence to flower at the shoot apical meristem. So first and foremost, you have to understand this that the plants they differ considerably in the age at which they become sensitive to vernalization. It is not that at any age you can initiate flowering. Okay, they have to reach a particular age. The age is very important. Now, winter annuals such as the winter forms of cereals, they respond to low temperature very early in their life cycle. So, in those cases, they can be vernalized before germination if the seeds have imbibed water and become metabolically active. But in case of biennials, they must reach a minimum size before they can become sensitive to low temperature for vernalization. So this clearly tells you that age is very important. All right. Now other plants, if you see, they including most biennials, they will form the rosette during the first season after sowing, and the flowering in the following summer. Okay. So that is why it is said that they need a minimal size before they can become sensitive to the low temperature. Now, what is this low temperature? There has to be some range. So it can be below freezing to about 10 degrees with a broad optimum usually between 1 to 7 degrees. This has been established by Lang in the year 1965. And the effect of cold increases with the duration of cold treatment until response is saturated. So this effective temperature for vernalization, this is from just below the freezing to about 10, right? So, you know, they usually require several weeks of exposure to low temperature, but the precise duration varies widely with species and variety. Now, this is not everything. Vernalization, cold temperature is just not everything. Vernalization requires photoperiod as well. So don't think that I can leave out photoperiod and I can only go with vernalization and then I will get flowering. No, that will not happen. So vernalization requirement, this is often linked with requirement for a particular photoperiod. And it has been seen that the most common combination is requirement for cold treatment followed by a requirement for long days. And if you do not devernalize it, this vernalized medicine can remain competent to flowering for as long as 300 days, even in the absence of inductive fertility. That means when you are inducing the meristem, that time you need both vernalization and photoperiod. Okay, but once you have induced it, then even if you don't give the photoperiod, And if you don't de-vernalize this induced condition will remain for a very, very long time. Okay? So what is the gene that is causing all of this? So it has been shown by Mikhail, who is a Russian scientist, postulated the existence of universal flowering hormone, which is known as florigen, to be behind all this induction. So this FT protein produced by the genes flowering locus T in Arabidopsis and the corresponding proteins in other species may be well an important part of the florigen. Now, of course, if there is something that promotes this flourishing production, there will be some genes which is like inhibiting the flourishing production. Okay, and what is important here is this flourishing. This is controlling the entire thing. Now, as we have already said that if your plant has been induced, it will stay for a longer time, and not only that, under photoperiod, we have already seen that if you can induce the leaf, if you take out a leaf, the leaf can be induced, right? And that induction can be transmitted to another plant. So from these graft experiments, it has been seen that if you induce a plant, you take a graft of this plant, induced plant, you graft it onto an uninduced plant. What will you see? That this florigen will get transmitted. Remember, we said what? That the floral signal is a chemical type. So, what is that chemical? It is a hormone. It is the florigen. So, that will get transmitted. Now, this branch was a part of the uninduced plant. Now, how can you prove that this florigen is getting transmitted? If you take a graft of this, okay, this section, and plant it to another plant, uninduced plant, here what will you see? That here also this plant is getting induced. Okay, so if you take this, you plant it here, what is happening? It is getting induced. If you take this, plant it here, this section is getting induced. If you take this, plant it here, the rest of it is getting induced. The florigen is here. Okay, now this is what is known as an indirect induction. Why it is an indirect induction? Because here the young leaves are developed on the receptor plant. Alright, after it had been induced to flower by a donor leaf. This is your donor leaf, this is your receptor leaf. They can do what? They can themselves be used as donor leaves in the subsequent grafting experiments. So that is why we call this as indirect induction. Okay. Now in this case, remember the strength of this floral stimulus from the donor leaf to your recipient leaves. This remains constant even after serial grafting. So whatever uh, strength it was here, the same strength is maintained here. The same strength is maintained here, here, and here. Okay, so that is why we can say that uh, the induced state is in some way propagated through the plant. Now, in contrast to this direct induction, there is another kind of induction, which is known as your uh, this is your uninduced one. Okay, this is an indirect induction. So this is seen in the case of your perilla, whose only leaf you are taking for inductive period. So you are only taking a single leaf. You are inducing the single leaf. You are grafting the single leaf on an uninduced stalk. You will find this uninduced stalk is also getting the signal. Again, if you take the same leaf from here and plant it on another uninduced stalk, again you will see that this uninduced stalk is getting the same. Again, if you take the same leaf and plant it on another graph, again this will get induced. Okay, so just one leaf which has been induced can be used to induce many other uninductive stalks. Okay, so that is why we call it as indirect induction. It's not direct, it is via an induced portion, which can be a induced stalk, like in the previous case, or it can be a leaf. I hope this is clear. Okay, so these are the pointers that you have to remember for your exam point of view. The experiments are important, and these points which I have mentioned, this is very, very important. So that is all for today's discussion. I'm sure by now you have got an idea about what is photoperiodism and what is vernalization. Okay, the concept might look the same, but they can confuse you with different situations. So when you are learning this chapter, be very careful about the different situations which I have explained with the help of the graphs. Okay, so with this note, I will take a leave today. Thank you. Good evening, a good night, and bye bye. Wish you all the best.